A gene carries the information that determines your traits, which are features or characteristics that are passed on to you, which are inherited from your parents. Each cell in the human body contains about 25,000 to 30,000 genes. A gene mutation is a permanent alteration in the DNA sequence. Mutations range in size and can affect anywhere from a single DNA building block to a large segment of chromosomes that include multiple genes. There are two types of mutations. The first type are hereditary mutations, which are inherited from a parent and are present throughout a person's life in every cell in the body. The second type are somatic mutations, which occur at some time during a person's life and are present only in certain cells. These changes are caused by environmental factors like radiation from the sun. Gene patenting stirred up controversy in the 1970s with the U.S. Supreme Court case Diamond v. Chakrabarty. In this case, a scientist named Chakrabarty attempted to patent a bacterium that he had engineered specifically to consume hydrocarbons, but was stopped by the U.S. Patent Office. In the end, the court ruled that Chakrabarty could patent his bacterium because he had altered its genetic makeup from its natural state. This eventually led in the 80s to the allowance to patent human genes that had a known function and were altered in some way, or genes that were not entirely natural, referred to as complementary DNA, which was a step up from previously only being able to patent plant genes. Soon after scientists switched to patent unaltered human genes in the 1990s, and scientist J. Craig Venter attempted to patent human gene fragments from the brain called express sequence tags, or ESTs. This was rejected by the courts because they were not able to express genes with a certain known and definite function, such as the insulin gene, and it was later concluded to not be useful for research for patents. Still, the initial gene patenting case led to researchers, scientists, universities, institutions, and corporations to attempt to claim ownership and patent unaltered human genes. Most people claim they want to be able to patent genes in order to better understand the human genome and perform effective research on human genes. In addition, this can lead to genetic donors not getting any profit for their genes, as seen in the case of Henrietta Lacks. It also prompts the question, is it ethical for someone to have ownership over a part of another human being without their knowledge or consent? Today, the court holds that a naturally occurring DNA segment is a product of nature and not to be patented simply because it has been isolated. Association for Molecular Pathology vs. USPTO. In 2009, the American College of Medical Genetics, American Society for Clinical Pathology, College of American Pathologists, 3 other healthcare organizations, and several individuals sued the USPTO, Myriad Genetics, and the director of the University of Utah Research Foundation in the United States District Court for the Southern District of New York. The suit challenged the validity of the patents on the BRCA1 and BRCA2 breast cancer susceptibility genes, claiming that the patents are overly broad and in conflict with policies that prohibit the patenting of natural phenomenon and basic human knowledge and thought. In 2010, a federal court ruled that several of the BRCA gene patents were invalid. Myriad Genetics appealed the case to the United States Court of Appeals for the Federal Circuit. In July 2011, the Court of Appeals reversed the federal court decision, ruling that the genes were patentable. The patents allowed Myriad to control access to the genes, known as BRCA1 and BRCA2, and gave the company the right to limit others from doing research or diagnostic testing of the genes. Opponents of the patents argued that research and diagnostic testing from others is crucial for patients making important medical decisions. They also said that the patents allowed Myriad, which sells the only BRCA gene tests, to set the costs and other parameters of tests, making it difficult for women to access alternate tests or get a comprehensive second opinion about the results. After reconsideration of the case, the Court of Appeals upheld its decision. In April 2013, the U.S. Supreme Court considered the case. In June of 2013, the Supreme Court ruled that naturally occurring genes are not patentable. invalidating the patents held on BRCA1 and BRCA2. In January 1951, a woman by the name of Henrietta Lacks was diagnosed with cervical cancer at Johns Hopkins Hospital in Baltimore, Maryland, and would die later that year because of it. The cells taken from Henrietta, without her permission, were then passed on to a scientist named George Otto Gay, who would go on to create the first human immortal cell line, later known as HeLa cells. The ability of HeLa cells to rapidly reproduce led to many important breakthroughs in biomedical research, specifically with polio, cancer, AIDS, and the effects of radiation and toxic substances. Neither Henrietta Lacks nor her family gave researchers permission to harvest her cells, which at the time permission was not legally needed. Since 1955, over 10,000 patents have been registered involving HeLa cells, with the Lacks family not receiving any part of the profit. Along with legal concerns of gene patenting are ethical concerns which many bioethicists, philosophers, theologians, scientists, and researchers have voiced. Many argue that by allowing gene patenting and applying patented augmentation genes interferes with the natural order of nature and that we are playing God. Arguments against this concern have pointed out that because human beings have rationality, which separates us from animals, a capacity to make normative judgments and act on reason, we will further improve our capacity to utilize these newly discovered features. The most powerful concern about gene patenting is universal access, which currently is only attainable for those who are wealthy. Because the wealthy will be the first to have access to augmented gene technologies, it will spawn the ultimate inequality, a genetic caste system. We are strongly against gene patenting due to ethical concerns, amongst others. For instance, gene patents impose many limitations and restrictions that would harm future research and development, has limited access to all, and too costly for patients. In the court case, Molecular Pathology vs. Myriad Genetics, the Supreme Court unanimously agreed that human genes may not be patented. However, there still remains incentives for discovering these genes, as they bring more potential than just a simple discovery. Researchers and scientists may manipulate genes to fix or alter their function, which is legally patentable. This may lead to various uses for treating these discovered genes, such as gene therapy. The many possible applications for discovering these genes justifies the removal of gene patents.